Welcome to the Deep Dive, the show where we really try to pull back the curtain on complex subjects, sift through the information, and, extract the most important nuggets so you walk away truly well-informed. Today, we're diving into one of your body's most incredible feats of metabolic engineering, a real behind-the-scenes hero. Have you ever wondered how your brain stays razor-sharp, your red blood cells keep pumping, and your muscles can still function, even when you haven't eaten a single carbohydrate for hours, maybe even days? That's right, even on an empty stomach, your body had this, ingenious, masterful way of making glucose pretty much from scratch. Okay, let's pull back the curtain on this. Our mission today is to truly dissect a complex yet absolutely vital pathway known as gluconeogenesis. Think of it as your body's ingenious backup factory for glucose production, kicking into high gear when dietary carbohydrates are scarce. We're going to ensure you understand not just what it is, but how it works, why it's such a lifesaver, and who, meaning which organs and molecules, are the essential players in this remarkable process. We've brought together a fantastic set of sources that truly lay out the intricate details, and we're ready to explore them with you, helping you master this fundamental biological process.
Yeah, and what's truly remarkable here is just how critical a continuous supply of glucose is for specific, absolutely non-negotiable tissues in your body. We're talking about your brain, of course, which is a major greedy consumer of glucose, but also your red blood cells. They famously lack mitochondria, right? So they rely entirely on glucose breakdown for their energy.
Right. No other choice for them.
Exactly. Then there's the kidney medulla, the lens and cornea of your eye, the testes, and even your exercising muscle. All of these absolutely depend on glucose as their primary, or in some cases, their sole metabolic fuel. Now, your liver stores glycogen, which is, a fantastic short-term source of glucose, especially after a meal.
But short-term is the key word there.
It is just that short term. Typically, those stores last less than 24 hours if you're not taking in any carbs. Once that hepatic glycogen is depleted, your body faces a critical moment. Glucose must be synthesized from non-carbohydrate precursors. It's not optional. It's a fundamental shift, really a survival mechanism. And it's crucial to understand this isn't simply glycolysis, the glucose breakdown pathway, just running in reverse.
Ah, okay. That's a common misconception, maybe.
It is. The energetics are fundamentally different. Glycolysis strongly favors pyruvate formation, so a special, distinct pathway, gluconeogenesis, is absolutely necessary to push the reaction in the opposite direction towards making glucose.
So if gluconeogenesis is literally building glucose from scratch from things that aren't carbs, well, that sounds like a pretty monumental task. Is it all happening in one place, or is this more of a distributed effort across the body?
That's a great question because it's definitely not a single location operation, not at all. At its core, gluconeogenesis is the de novo, or from new, synthesis of glucose from those non-carbohydrate precursors we mentioned. But what's really fascinating is its dual location nature. It requires specific enzymes found in both the mitochondria, those cellular powerhouses, and in the cytosol, which is this sort of jelly-like fluid filling the cell.
So two different compartments inside the cell need to cooperate.
Precisely. You can genuinely think of it like a highly specialized manufacturing plant. Imagine you have different assembly lines, each with its own specific machinery, but these lines are actually spread across two distinct buildings, one in the mitochondrial building and another in the cytosolic building. They have to work in concert, passing intermediates back and forth to ultimately produce that final glucose product. It's a complex, coordinated dance.
The metabolic dance, huh? So which parts of our bodies are hosting this incredible glucose factory? Who are the main manufacturers here?
Right. Well, if we connect this to the bigger picture of glucose production, the liver is absolutely the main player, no question. Especially during something like an overnight fast, it contributes the dominant share, around 90% of the newly synthesized glucose.
90%? Wow, that's huge.
It is huge. However, it's a dynamic system. It adapts to the body's needs. During prolonged fasting, and we're talking about extending beyond that initial 24-hour mark where liver glycogen is gone, the kidneys step up significantly. They actually become major glucose producers, contributing up to maybe 40% of the total glucose production.
So the kidneys take on a much bigger role over time.
A much bigger role, yes. And while its role is minor, the small intestine also plays a part in glucose synthesis, though it's less significant. The clinical relevance here is pretty profound. If there are deficiencies in any of the key enzymes involved in gluconeogenesis, the body's ability to maintain a stable blood glucose level is severely compromised.
Which leads to problems.
Oh, absolutely. It leads to hypoglycemia or dangerously low blood sugar, which can have severe consequences for the brain and those other glucose-dependent tissues we talked about. As our source material beautifully illustrates, this pathway isn't just some side alley in metabolism. It's one of the essential foundational pathways of energy metabolism. It's intricately woven into the entire metabolic network to ensure overall energy balance and, well, ultimately survival.
Okay, so it's crystal clear we need this glucose, especially when stores run low. But if our body's building it from basically zero, what are the actual raw materials? What are these non-carbohydrate precursors that power this whole operation?
Yeah, that's the beauty of the body's resourcefulness, isn't it? It primarily turns to three main categories of precursors when carbs are scarce. The most important gluconeogenic precursors are glycerol, which perhaps surprisingly comes directly from fats.
From fats, interesting.
Yep. Then lactate, the byproduct often associated with intense exercise.
Ah, the muscle burn molecule.
That's the one. And then a whole host of things called alpha keto acids, which are derived from the metabolism of what we call glucogenic amino acids. And here's a truly surprising fact that really highlights the body's incredible adaptability. Almost all amino acids, all but two, leucine and lysine, are actually glucogenic.
Wait, nearly all amino acids can become glucose.
Almost all of them. It means that if you break down proteins, their carbon skeletons can be converted essentially directly into glucose. It's quite amazing.
Okay, let's definitely delve into these raw materials then. Starting with that fat-derived fuel you mentioned, how exactly does glycerol get turned into glucose?
Absolutely. First up, glycerol. This is released during the breakdown of triacylglycerols, or TAGs, which are basically the main form of fat stored in your adipose tissue, your fat cells. Once released, glycerol, being water-soluble, just travels through your bloodstream, making its way primarily to the liver.
So the liver picks it up.
The liver picks it up. Inside the liver, it undergoes a quick two-step chemical makeover. First, an enzyme called glycerol kinase adds a phosphate group to it, creating glycerol 3-phosphate. Then another enzyme, glycerol 3-phosphate dehydrogenase, oxidizes this glycerol 3-phosphate, transforming it into dihydroxyacetone phosphate, or DHAP.
DHAP.
That sounds familiar.
It should. What's cool about DHAP is that it's a versatile middleman molecule. It's a direct intermediate in both glycolysis, the glucose breakdown pathway, and gluconeogenesis, the glucose synthesis pathway. This is a brilliant way the body taps into its vast fat stores for a glucose building block. Even though the fatty acids themselves, the long chains, cannot directly become glucose.
Right, okay. That distinction is important. So next up, lactate. Many of us might recognize that, like you said, from that familiar burning sensation during a really intense workout. How does lactate, often seen as just waste, get recycled into precious glucose?
That's right. Lactate isn't just waste. It's actually a critical signaling molecule and a really valuable precursor. It's produced during anaerobic glycolysis.
Meaning without enough oxygen. Exactly. Glucose breakdown without sufficient oxygen. This happens particularly in intensely exercising skeletal muscle, and also by cells that famously lack mitochondria, like your red blood cells. These cells produce a lot of lactate and just release it into the blood. Your liver then picks up this lactate, oxidizes it back to pyruvate.
Okay, so it reverses that step.
It reverses that step.
Okay.
And then, through the gluconeogenesis pathway we're discussing, it converts that pyruvate into fresh glucose. This newly synthesized glucose is then released back into the circulation where it can be taken up and used by the very muscles or red blood cells that produce the lactate in the first place.
Wow, that's a neat cycle.
It's a beautiful intertissue communication loop known as the Cori cycle. You can see its elegance illustrated in the source material. It's essentially your body's brilliant recycling program for those intense moments, ensuring that byproducts are efficiently converted back into fuel. And the process is incredibly efficient, Specialized transport proteins smoothly facilitate the diffusion of lactate and glucose across cell membranes.
Okay, Cori cycle. Got it. And finally, that third major source of raw material, especially when we're talking about prolonged periods without food, that comes from amino acids. How do our proteins contribute to making glucose?
Indeed. Amino acids are a crucial source, primarily derived from the breakdown of your own tissue proteins, which happens, for instance, during a fast when other fuels are low. When these proteins are broken down, their individual amino acids undergo metabolism that generates various carbon skeletons. These are often called alpha keto acids.
Like pyruvate again?
Exactly. Some amino acids yield pyruvate directly, which can be converted straight into glucose. Others, like maybe aspartate or glutamate, yield intermediates of the tricarboxylic acid cycle, the TCA cycle.
The Krebs cycle.
Yes, the Krebs cycle. Things like alpha ketoglutarate or oxaloacetate, OAA. These TCA cycle intermediates can then be directly funneled into the gluconeogenic pathway. OAA in particular is a direct and crucial precursor for phosphenol pyruvate, or PEP, which is a key intermediate we'll talk more about. Now, here's a really crucial distinction, one that often trips people up and is, frankly, vital for mastering this topic.
Okay.
Acetyl coenzyme A, or acetyl-CoA,
Okay.
and any compounds that only give rise to acetyl-CoA like acetoacetate, a ketone body, or the amino acids lysine and leucine, these cannot be used for a net synthesis of glucose.
Cannot. Why not?
It's a fundamental principle in metabolism. The enzyme complex that converts pyruvate to acetyl-CoA, the pyruvate dehydrogenase complex, or PDHC, is physiologically irreversible. Under normal body conditions, it only goes one way. This means that once pyruvate has been committed to becoming acetyl-CoA, there's no going back, no direct path to convert those acetyl-CoA carbons into pyruvate, and thus no direct path to glucose from them.
So those carbons are sort of locked out of glucose production.
Locked out of net glucose production, yes. These ketogenic compounds, as they're called, instead give rise to ketone bodies, which are important alternative fuels, particularly for the brain, during prolonged fasting. But they do not contribute net carbons to glucose synthesis. Now, this raises an important question. If acetyl-CoA cannot be directly converted to glucose, how can fats, which primarily break down into acetyl-CoA, still be considered a significant fuel for gluconeogenesis, even indirectly?
Right. That seems contradictory.
The answer, as we touched on, lies partly in that glycerol portion of the fat molecule, which can be converted to glucose, and as we'll soon discover, acetyl-CoA plays an absolutely critical regulatory role, kind of directing other carbon atoms towards glucose synthesis, even if it's not a direct building block itself.
Okay, here's where it gets really interesting, I think. You mentioned glycolysis is like a road trip, and gluconeogenesis is trying to go back the other way. Most of the reactions on this road are like two-way streets. They're reversible, meaning they can be used in reverse for gluconeogenesis. You said there are 7 of these.
7 reversible reactions, yes, that both pathways share. They use the same enzymes, just running in opposite directions depending on the conditions.
However, there are those 3 critical junctions, 3 specific reactions in glycolysis that are essentially one-way streets. They're physiologically irreversible. So to synthesize glucose, your body can't just drive backwards through these points. It needs special bypass roads, right?
Exactly, special bypass roads. These bypass reactions are unique to gluconeogenesis and are specifically designed to energetically favor the synthesis of glucose, allowing the metabolic traffic to flow in the uphill direction, so to speak.
And the irreversible glycolytic enzymes we need to bypass are pyruvate kinase, phosphofructokinase 1, and hexkinase or glucokinase.
Those are the 3 roadblocks, precisely. Each one requires a clever workaround, a unique detour catalyzed by different enzymes specific to gluconeogenesis.
Okay, let's tackle the first bypass. How do we get around that pyruvate kinase stack, going from pyruvate back up to PP?
That's a perfect analogy about the roadblock. The first major one we encounter is indeed that irreversible step catalyzed by pyruvate kinase, which in glycolysis converts PE down to pyruvate. In gluconeogenesis, we need to go the other way, from pyruvate up to PEP, and this requires a clever 2-step bypass. The very first step occurs deep within the mitochondrial building of liver and kidney cells. Here, pyruvate is carboxylated, meaning a carbon dioxide molecule is added to it, by the enzyme pyruvate carboxylase, or PC. This forms oxaloacetate, OAA.
Pyruvate carboxylase. Okay, does it need anything special to work?
It absolutely requires a helper molecule, a coenzyme called biotin. Biotin is actually tightly bound to the enzyme itself. It's essential for grabbing that CO2. The reaction itself is an energy-intensive process driven by the energy released from ATP hydrolysis. Specifically, ATP helps form a temporary intermediate involving the enzyme, biotin, and carbon dioxide, with bicarbonate in the cell providing that CO2.
And this enzyme, PC, you said it has a dual purpose.
Yes, and this is vital to understand. First, its primary role in this context is allowing the production of OAA, which is that key intermediate on the pathway towards PEP, absolutely crucial for gluconeogenesis. But second, it also provides OAA that can replenish the intermediates of the TCA cycle, especially if they get depleted during active metabolism. This is often called an anaplerotic role, meaning filling up.
So it keeps the TCA cycle topped up too.
Exactly. It's worth noting that muscle cells also contain pyruvate carboxylase, but in muscles, the OAA produced is only used for this replenishment purpose for the TCA cycle. They don't use it for glucose synthesis to release into the blood.
Ah, Okay, only liver and kidney do that.
Primarily, yes. And pyruvate itself needs help getting into the mitochondria. There's a specific transport protein for that. And this enzyme, PC, is actually part of a family of carboxylases that all require biotin, highlighting a common essential role for this vitamin in various metabolic pathways.
How is this step regulated? You mentioned regulation is key.
Right. PC activity is intricately regulated. It's allosterically activated by acetyl-CoA.
Activated by acetyl-CoA, the molecule that can't become glucose.
The very same. This is a critical control point, a precise signal. When acetyl-CoA levels are elevated in the mitochondria, which happens, for example, during fasting when fats are being broken down rapidly, it signals a metabolic state where there's an increased need for OAA synthesis because that OAA is being heavily used for gluconeogenesis in the liver and kidneys.
Makes sense. So high acetyl-CoA means make more OAA for glucose.
Exactly. Conversely, if acetyl-CoA levels are low, pyruvate carboxylase is largely inactive. In this scenario, pyruvate is primarily shunted towards oxidation by that other complex, the pyruvate dehydrogenase complex, to produce acetyl-CoA, which can then be burned in the TCA cycle for immediate energy. The body isn't prioritizing glucose synthesis then.
Wow, that's elegant.
Yeah.
One molecule directing traffic based on fuel availability.
It's beautiful metabolic fine-tuning.
Okay, so once oxaloacetate, OAA, is formed in the mitochondria, it's a crucial stepping stone. But you said it happens in the mitochondria, and the rest of the pathway needs to happen in the cytosol, but OAA can't just waltz out. That sounds like a problem.
It is a bit of a logistical challenge, yes. The inner mitochondrial membrane doesn't have a direct transporter for OAA, so the body uses a clever shuttle service.
A shuttle. How does that work?
OAA is first temporarily changed into another molecule, malate. This transformation is catalyzed by an enzyme called mitochondrial malate dehydrogenase. Critically, this malate can be transported out of the mitochondria into the cytosol via a specific transporter.
Okay, so OAA becomes malate, gets right out, then what?
Once in the cytosol, it's immediately changed back, reoxidized back to OAA by a cytosolic version of malate dehydrogenase. And here's why the shuttle is so brilliant beyond just transport. This reoxidation process in the cytosol also reduces NAD+ to NADH.
Generating NADH in the cytosol.
Exactly. And this cytosolic NADH is absolutely crucial because it's needed later in gluconeogenesis, specifically for a reduction step involving 1,3-bisphosphoglycerate being converted to glyceraldehyde 3-phosphate, a reaction that's common to both glycolysis and gluconeogenesis, but running in reverse here.
So the shuttle doesn't just move the carbons, it also moves the reducing power needed later.
Precisely. It ensures the necessary reducing power, NADH, is available in the right compartment, the cytosol, for glucose synthesis to continue. It's also worth noting the body has alternative routes, showing its robustness. If lactate is already abundant in the cytosol, maybe from that corticycle activity,
Right.
that lactate can be oxidized to pyruvate right there in the cytosol, and that reaction also produces cytosolic NADH.
So if you have lactate, you don't need the malate shuttle for NADH.
Not necessarily for the NADH part, no. In that case, pyruvate can enter the mitochondria, get converted to OAA by PC. Then that OAA can actually be converted to PAA by a mitochondrial version of the next enzyme, PPCK. And PP, unlike OAA, can be transported out to the cytosol directly.
Wow, multiple options.
Multiple options. OAA can even be converted to another amino acid, aspartate, which can be transported out and then converted back to OAO in the cytosol. These multiple routes just underscore the metabolic flexibility and redundancy built into the system to ensure glucose production continues under varied conditions.
Okay, that's intricate, but makes sense.
Okay.
So following the great mitochondrial escape of OAA, either as malate or PEP, or via aspartate, what's the final step in this first major bypass, the one that finally gets us from OAA or its equivalent to PE in the cytosol?
Right. Assuming OAA made it to the cytosol via malate or aspartate, the final step of this first major bypass involves its decarboxylation. OAA is both decarboxylated, meaning it loses a carbon dioxide molecule, and simultaneously phosphorylated to form PP. This is done by the enzyme p-carboxykinase, or PPK, operating in the cytosol, or sometimes the mitochondrial version, as we discussed.
PPK. And does this step need energy too?
It does. This reaction is energetically driven by the hydrolysis of guanosine triphosphate, or GTP, which is energetically similar to ATP. That GTP provides the necessary kick for this otherwise unfavorable conversion. So the combined actions of pyruvate carboxylase, PC, in the mitochondria using ATP and PPCK using GTP provide an overall energetically favorable pathway that completely circumvents that irreversible pyruvate kinase step from glycolysis.
Got it. PNO adds CO2, PPCK takes it off again, using energy along the way.
Essentially, yes. This allows the pathway to proceed smoothly from pyruvate all the way up to the next hurdle, which is fructose 1,6-bisphosphate, using those shared reversible reactions of glycolysis in between. And this pairing of carboxylation followed by decarboxylation is actually a brilliant biochemical strategy. It's used to drive reactions that would otherwise be energetically very difficult. You essentially invest energy to add the CO2 and then release that energy when you remove it to push the main reaction forward. We see this elegant mechanism in other crucial metabolic processes too, like fatty acid synthesis. It's a recurring, ingenious design principle.
Okay, bypass number 1 conquered. Moving on to the second major bypass. This one addresses the next big roadblock, which is that irreversible phosphofructokinase 1, or PFK1, reaction in glycolysis. How does the body get around this one?
For this bypass, gluconeogenesis employs a different enzyme, fructose 1,6-bisphosphatase, or FBPase1.
Fructose 1,6-bisphosphatase, so it just removes a phosphate.
Exactly. This enzyme, found primarily in the liver and kidneys, hydrolyzes fructose 1,6-bisphosphate, effectively snipping off one of the phosphate groups and yielding fructose 6-phosphate. This reaction is energetically favorable. It releases energy, ensuring the pathway continues to flow uphill towards glucose synthesis. It also represents another crucial regulatory site for gluconeogenesis, a key control knob for the whole pathway.
Another control point.
Yeah.
How is this one regulated?
The regulation of FBPase1 is quite sophisticated and really demonstrates beautiful metabolic control. First, it's exquisitely sensitive to the cell's own energy levels. If the cell's energy state is low, often signaled by a high ratio of adenosine monophosphate, AMP, to ATP.
High AMP means low energy.
Right. High AMP means ATP has been used up. In that state, fructose 1,46-bisphosphatase is inhibited. This makes perfect sense, doesn't it? Gluconeogenesis is an energy-requiring pathway. If the cell is already low on energy, it shouldn't be expending precious resources to synthesize glucose. It needs to conserve energy or make more.
Yeah, that's logical. Shut down the expensive project when funds are low.
Precisely. Conversely, when AMP levels are low and ATP levels are high, signaling abundant energy, gluconeogenesis is stimulated, allowing FBPase1 to work and the pathway to proceed. Second, and perhaps even more elegantly, FBPase1 is powerfully inhibited by a special little molecule called fructose 2,6-bisphosphate, not the 1,6 we just talked about.
Correct. A different molecule, fructose 2,6-bisphosphate. This acts as an allosteric effector, meaning it binds to the enzyme at a site other than the active site, effectively changing its shape and dialing down its activity. And the concentration of fructose 2,6-bisphosphate itself is very tightly controlled, influenced heavily by the insulin to glucagon ratio, that crucial hormonal signal we keep mentioning. Okay, how does glucagon affect this one?
When glucagon levels are high, which signals fasting, the enzyme responsible for making fructose 2,56-phosphate, it's a bifunctional enzyme called TFK2-FBPase2, but let's focus on its effect, is less active in making it. Consequently, less fructose 2,6-bisphosphate is present. Less fructose 2,6-bisphosphate means less inhibition of our gluconeogenic enzyme, fructose 1,6-bisphosphatase. So gluconeogenesis proceeds more robustly.
Wow. So glucagon lowers the inhibitor, which lets gluconeogenesis run.
Exactly. And here's the key concept again, reciprocal control. The signals that inhibit gluconeogenesis here, like high AMP or high fructose 2,6-bisphosphate, have the exact opposite effect on glycolysis. They activate PFK1, the corresponding glycolytic enzyme.
So it's like a metabolic seesaw. One goes up, the other goes down.
A perfect analogy. It provides this beautiful and precise balance between the pathways for glucose synthesis and glucose oxidation, preventing wasteful futile cycles where both might run simultaneously and just burn up ATP and GDP for no net gain.
Okay, that makes a lot of sense. So finally, we arrive at the third and last bypass. This deals with the irreversible hexokinase or glucokinase reaction from glycolysis, the very first step of glucose utilization where glucose gets trapped in the cell. What's the body's elegant solution to free up the newly made glucose at the very end of the line?
This final step in gluconeogenesis is handled by yet another unique enzyme, glucose 6-phosphatase.
Glucose 6-phosphatase. Sounds straightforward. Removes the phosphate from glucose 6-phosphate.
That's exactly what it does. This enzyme hydrolyzes glucose 6-phosphate, removing that phosphate group and providing a direct, energetically favorable pathway for the formation of free glucose. This free glucose is then ready to be released into the bloodstream. And the liver is the undisputed champion here. It's the primary organ responsible for producing free glucose from glucose 6-phosphate and releasing it into the bloodstream for other tissues, like the brain, to use.
But this isn't just a simple enzyme floating around, is it? I think I remember reading it's more complex.
You're absolutely right. It involves a rather sophisticated protein complex actually embedded within the membrane of the endoplasmic reticulum, or ER.
The ER, not the cytosol or mitochondria.
Yep, the ER membrane. This complex is composed of two main players. First, a glucose 6-phosphate translocase, which acts like a specialized ferry, transporting the glucose 6-phosphate across the ER membrane from the cytosol into the ER lumen. And second, the glucose 6-phosphatase enzyme itself, which is located inside the ER lumen, performs the actual phosphate removal there.
Why the complex setup in the ER?
That's a great question, and the exact reasons are still debated, but it likely helps regulate glucose release and prevents interference with other cytosolic processes involving glucose 6-phosphate like glycolysis or glycogen synthesis. This complex is found almost exclusively in gluconeogenic tissues, primarily the liver and kidneys, precisely where free glucose needs to be produced and released into circulation.
And it has a dual role, right?
Yeah, it does. What's truly fascinating is its dual role. It's not only essential for the final step of gluconeogenesis, but it's also absolutely required for the final step of glycogen degradation, glycogenolysis, breaking down stored glucose.
So both making new glucose and releasing stored glucose depend on this same final step.
Exactly. It highlights a critical convergence point in glucose metabolism. Clinically, deficiencies in either the phosphatase enzyme itself or the translocase protein lead to severe fasting hypoglycemia. These are known as glycogen storage diseases type IA and IBEM, respectively. This happens because, without these crucial proteins working, your body simply cannot produce free glucose from either making it new via gluconeogenesis or from breaking down its glycogen stores. The glucose 6-phosphate gets trapped, leading to dangerously low blood sugar during fasting.
That really underscores its importance. So once the free glucose is made inside the ER,
Once that free glucose is produced inside the ER lumen, specific transporters then move it out into the cytosol, and finally, other transporters like GLUT2 in the liver move it out of the cell and into the bloodstream where it can nourish your brain and other vital organs.
Okay, let's try to unpack this summary of the reactions because it really ties everything together and shows the body's metabolic ingenuity. Of the 11 reactions needed to convert 2 molecules of pyruvate all the way back up into 1 molecule of free glucose, 7 are actually catalyzed by those same reversible glycolytic enzymes, like those common two-way roads we talked about.
That's right. 7 shared reversible steps.
However, the 3 physiologically irreversible reactions from glycolysis, those catalyzed by hexokinase glucokinase, PFK1, and pyruvate kinase, they have to be deftly circumvented by 4 unique enzymes specific to gluconeogenesis.
Precisely. Those unique bypass enzymes are the key players. You've got pyruvate carboxylase, PC, converting pyruvate to oxaloacetate, needing biotin and ATP, and crucially activated by acetyl-CoA. Then oxaloacetate is converted to phosphenol pyruvate, PP, by carboxykinase, PPC, a reaction driven by GTP hydrolysis. The transcription of the PPCK gene itself is a major point of long-term regulation boosted by hormones like glucagon and cortisol during fasting or stress, and suppressed by insulin when you're fed.
Right, controlling the factory's capacity.
Exactly. The second bypass involves fructose 1,6-bisphosphatase, FBPase1, converting fructose 1,6-bisphosphate down to fructose 6-phosphate. This enzyme is inhibited by high AMP, signaling low energy, and by fructose 2,6-bisphosphate, which fascinatingly is also the primary allosteric activator of glycolysis. Perfect reciprocal control.
That's seesaw again.
That seesaw again. And the final bypass uses glucose 6-phosphatase, which dephosphorylates glucose 6-phosphate to release free glucose. This enzyme, located within the ER membrane as part of that complex, catalyzes the final step for both gluconeogenesis and glycogen breakdown. Its deficiency causes that severe fasting hypoglycemia, highlighting its critical role.
Okay, and this brings us squarely to the energy cost, which seems really important for understanding why this pathway needs such tight regulation. Building glucose from scratch sounds energy intensive, and it absolutely is, right?
It absolutely is.
Yeah.
The stoichiometry, the accounting of gluconeogenesis, starting from 2 pyruvate molecules, is quite significant. It requires the cleavage of a total of 6 high-energy phosphate bonds.
6.
6. Specifically, 4 ATP molecules and 2 GTP molecules are consumed. Additionally, it requires the oxidation of 2 NADH molecules using up reducing power. All of this energy investment culminates in the formation of just 1 single molecule of glucose.
Wow. 6 ATP-DTP equivalents and 2 NADH for 1 glucose.
Exactly. What's truly remarkable here and important to grasp is that this clearly demonstrates that gluconeogenesis is an energy-requiring or endergonic process. It consumes energy. It doesn't produce it like glycolysis does. This is precisely why its regulation is so vital. The body needs to ensure that glucose is only synthesized when it's absolutely necessary, like during fasting, and crucially, only when ample energy resources, typically coming from fat breakdown, are available to actually power the process. It's expensive metabolically.
Okay, now let's really zoom in on that regulation because, like you said, a pathway this complex and energy-intensive isn't just left to run wild. Your body has these incredibly sophisticated control mechanisms to ensure glucose is made precisely when and where it's needed, and importantly, only then. What are the main ways this is managed?
You're absolutely right. Control is paramount. There are basically two main types or levels of regulation at play here. First, there's the rapid moment-to-moment control. This is primarily dictated by things like circulating glucagon levels, the fasting hormone, and also the immediate availability of those raw materials, the substrates like amino acids and lactate.
So immediate signals and fuel supply.
Exactly. And then there are slower, more adaptive changes. These involve altering the actual amount of the key enzymes involved, usually by changing the rate at which their genes are transcribed and translated into protein, so either increasing their synthesis or decreasing their degradation. This essentially changes the maximum capacity of the glucose factory itself over hours or days.
Adjusting the machinery size, basically.
That's a good way to put it. And the broader hormonal control of this whole glucoregulatory system involving hormones like insulin acting opposite to glucagon, plus others like cortisol. It's a truly fascinating topic in itself, a masterclass in metabolic orchestration, really.
Okay, let's zoom in on glucagon first. We've heard it mentioned quite a few times now as the fasting signal. How exactly does it push gluconeogenesis into overdrive when blood sugar is low?
Glucagon, that peptide hormone secreted by the alpha cells of your pancreatic islets, is arguably the most significant hormonal signal for ramping up gluconeogenesis during fasting. It stimulates glucose synthesis through really three distinct yet interconnected mechanisms. First, it directly changes the levels of those crucial allosteric effectors we discussed. Remember fructose 2006-bisphosphate?
The inhibitor of FP-PACE1.
Exactly. When glucagon levels are high, it causes a reduction in the hepatic levels of fructose 2-6-bisphosphate. As we discussed, less of this inhibitor means that fructose 1-6-bisphosphatase, our gluconeogenic bypass enzyme, is activated. Simultaneously, phosphofructokinase 1, the key glycolytic enzyme, is inhibited. This neatly tilts that metabolic seesaw we talked about, powerfully favoring glucose synthesis over glucose breakdown in the liver.
Okay, so it removes the brakes on gluconeogenesis and applies them to glycolysis.
Perfectly put. Second, glucagon also exerts control through covalent modification of enzyme activity, basically, adding or removing phosphate groups to switch enzymes on or off. Glucagon binds to its specific receptor on the liver cell surface. This binding kicks off a signaling cascade inside the cell involving KMP, which ultimately activates an enzyme called protein kinase A, or pKa. pKa then tags hepatic pyruvate kinase, remember, one of those irreversible one-way glycolytic enzymes, with a phosphate group. This phosphorylation effectively turns pyruvate kinase off.
Turning off the last step of glycolysis.
Yes. This inactivation of pyruvate kinase means that PEPP, phosphenol pyruvate, instead of being broken down to pyruvate, is now diverted upwards, straight into making new glucose via gluconeogenesis. And it's important to note, this specific covalent regulation of pyruvate kinase only happens with the liver isozyme, ensuring this control happens specifically in the liver, the main organ for releasing glucose systemically.
Targeted control, very neat. What's the third mechanism?
The third way glucagon boosts gluconeogenesis is slower but potent. It can actually induce the synthesis of more enzyme molecules. This is a longer-term adaptive response. Glucagon directly increases the transcription of the gene for PPI carboxykinase, PPCK, via a specific transcription factor called CREB.
So it tells the cell's DNA to make more PPCK.
Essentially, yes. This means that during prolonged fasting, as the need for glucose production continues, the liver cell actually produces more of the PPCK enzyme itself, physically increasing its capacity for gluconeogenesis. And as a side note, the stress hormone cortisol also increases PPCK gene expression, synergizing with glucagon, while insulin, the hormone of the fed state, actively decreases its expression. Beautiful reciprocal control right down to the genetic level.
That's amazing control. Now, beyond these powerful hormonal signals like glucagon, the simple availability of the raw materials themselves, those substrates, must be absolutely critical too, right? You can't build something with no bricks.
Absolutely critical. The presence of ample gluconeogenic precursors, especially those glucogenic amino acids, directly influences how fast your body can actually synthesize new glucose. The enzymes might be ready, but they need fuel. For instance, when insulin levels decrease, which is the natural hormonal response during fasting, this favors the mobilization of amino acids from muscle protein. Essentially, the body starts breaking down a small amount of muscle to release amino acids.
So fasting signals muscle to release building blocks.
In a controlled way, yes. These released amino acids then provide the essential carbon skeletons that the liver and kidneys can grab and use for gluconeogenesis. And think about all that energy required for this complex endergonic process, the ATP and NADH coenzymes we calculated earlier. Where does that come from during fasting?
From fat breakdown.
Primarily, yes. It's mainly supplied by the oxidation of fatty acids flowing from adipose tissue. This highlights how fat breakdown is crucial, not just for providing direct energy for the rest of the body, but specifically to fuel the energy-intensive process of glucose synthesis in the liver and kidneys when carbohydrate stores are low. It's a beautifully integrated system. Fat burns to power glucose synthesis from amino acids and glycerol.
Right, it all connects. Now, this brings us to another fascinating regulatory point you mentioned earlier, that allosteric activation of hepatic pyruvate carboxylase, PC, by acetyl-CoA. This seems like a really subtle point, but why is it so important, especially during fasting?
It might seem subtle, but this mechanism becomes particularly prominent and powerful during fasting, and it's a truly brilliant example of metabolic traffic direction. Here's why. During a fast, there's that increased breakdown hydrolysis of triacylglycerols in your fat cells. This floods the liver with fatty acids. The liver then rapidly oxidizes these fatty acids through beta oxidation, producing a large amount of acetyl-CoA. Often, this rate of acetyl-CoA formation exceeds the liver's capacity to oxidize it completely to carbon dioxide and water through the TCA cycle.
So acetyl-CoA starts to pile up.
Exactly. Acetyl-CoA accumulates inside the mitochondria. This accumulating acetyl-CoA then acts as that positive allosteric activator of pyruvate carboxylase, PC, effectively signaling, hey, we've got plenty of fat fuel coming in, enough energy to power glucose synthesis, so let's get making OA.
Okay, so it activates the first step towards glucose.
Yes, but here's the truly elegant part, the key concept that ties it together. Acetyl-CoA also has another crucial role simultaneously. It actively inhibits the pyruvate dehydrogenase complex, PDHC.
The enzyme that converts pyruvate to acetyl-CoA.
The very one. Acetyl-CoA inhibits PDHC, partly by activating an enzyme, PDH kinase, that phosphorylates and inactivates PDHC. So, think about it. This single compound, acetyl-CoA, acts as a master conductor, strategically directing the fate of pyruvate. It effectively diverts pyruvate away from being oxidized by PDHC for immediate energy and towards carboxylation by PC for gluconeogenesis.
Wow, so it blocks one road and opens another for pyruvate.
Precisely. It truly raises the question. How can one relatively simple compound, acetyl-CoA, act as such a precise and multifaceted metabolic traffic director for pyruvate? It ensures that during fasting, precious pyruvate, coming from lactate or amino acids, is preferentially used for vital glucose synthesis rather than just being burned for immediate energy that's already being supplied by fats. It's a remarkable example of metabolic efficiency and coordination.
That is remarkable. Okay, and finally, let's circle back quickly to that other allosteric regulator, AMP. How does this molecule act as an...
Right. AMP, adenosine monophosphate, serves as a very direct sensor of the cell's immediate energy charge or status. Fructose 1.6 bisphosphatase, that key gluconeogenic enzyme in the second bypass, is powerfully inhibited by AMP. And remember our seesaw. This is another brilliant example of reciprocal regulation because AMP is also a compound that activates phosphofructokinase 1, PFK1, the corresponding rate-limiting enzyme in glycolysis, which breaks down glucose to make energy.
So high AMP inhibits glucose synthesis and stimulates glucose breakdown.
Exactly. When AMP levels are elevated, it's a clear biochemical distress signal to the cell that energy levels are critically low, meaning ATP has been heavily used up and broken down first to ADP and then to AMP. In response, the body logically stimulates energy-producing pathways like glycolysis to rapidly generate more ATP, and simultaneously it inhibits energy-requiring pathways, such as gluconeogenesis, which would only consume precious ATP and GTT.
Makes perfect sense. Prioritize making energy when you're running low.
It's a perfectly orchestrated system that ensures metabolic harmony, prioritizing immediate energy production when reserves are low, and halting expensive energy-consuming projects like glucose synthesis until the energy situation improves.
Okay, so we've really journeyed through this incredibly intricate pathway. Let's try to synthesize everything we've covered now, really reinforcing the key takeaways for mastery. First, those main raw materials, the precursors your body uses. Can you quickly recap those for us one more time?
Absolutely. Let's nail these down. The main gluconeogenic precursors are, first, glycerol, which comes from the breakdown of those triacylglycerols in your fat tissue. Second, lactate, the byproduct from cells lacking mitochondria like red blood cells or from intensely exercising skeletal muscle via the Cori cycle. And third, the alpha-keto acids, such as alpha-ketoglutarate and especially pyruvate, which are derived from the metabolism of glucogenic amino acids, primarily coming from protein breakdown during fasting. And remember, 7 of the reactions that convert pyruvate back up to glucose are those reversible glycolytic enzymes, the shared two-way streets. But the 3 crucial irreversible steps from glycolysis must be bypassed by unique enzymes found only in gluconeogenesis.
Right. And those unique bypass enzymes are really the heroes of this story, aren't they? Could you quickly walk us through the function of each one again, just highlighting their absolute significance?
Indeed, they are the key players. First, we have pyruvate carboxylase, PC. It converts pyruvate to oxaloacetate in the mitochondria, requires biotin as a cofactor, uses ATP for energy, and importantly, it's allosterically activated by acetyl-CoA, signaling that fat breakdown is active and energy is available. Then, oxaloacetate, or its equivalent, is converted to phosphenolpyruvate, PP, by PP carboxykinase, PC. This reaction uses G2-key hydrolysis for energy, and the expression of the PPCK gene itself is heavily regulated by hormones like glucagon up and insulin down, controlling the pathway's long-term capacity.
Got it. PC and PPCK for the first bypass. What about the second?
The second bypass uses fructose 1,6-bisphosphatase, a PPS1. This enzyme converts fructose 1,6-bisphosphate down to fructose 6-phosphate. It's inhibited by high AMP, signaling low cellular energy, and by fructose 2,6-bisphosphate, the molecule whose levels are controlled by glucagon insulin, providing that elegant reciprocal control with glycolysis.
Okay, FP-PACE1 for the second bypass and the final one.
The final bypass employs glucose 6-phosphatase. This enzyme dephosphorylates glucose 6-phosphate to release free glucose. It's located within the endoplasmic reticulum membrane as part of that transport complex, and critically, it catalyzes the final step for both gluconeogenesis and glycogen degradation. Its deficiency, as we emphasized, leads to severe fasting hypoglycemia because the body can't release glucose from either source.
That really locks in their roles.
Yeah.
Now let's tackle some of those deeper insights and maybe common misconceptions again, because truly mastering this means understanding the why and the implications, not just the steps.
Absolutely. What's truly crucial here, let's reiterate. Gluconeogenesis is absolutely an energy-requiring endergonic pathway. It consumes significant amounts of ATP and GTP. It does not produce net energy. This directly contrasts with glycolysis, which has a net release of ATP. Its vital role is in maintaining blood glucose levels, particularly during prolonged fasting. Think a 2-day fast or longer, once your readily available liver glycogen stores are completely depleted. That's when it really becomes life-sustaining.
And it's turned on by fasting signals.
Exactly. It's stimulated by a fall in the insulin to glucagon ratio, which is the key hormonal signal indicating a metabolic shift from the fed state to the fasting state, telling the liver and kidneys it's time for internal glucose production. And remember its location. It primarily occurs in the liver, mostly, and the kidneys, increasingly important during prolonged fasts. And it utilizes enzymes in both the mitochondria and the cytosol. Crucially, it does not occur significantly for glucose export in the cytosol of muscle cells. Muscle uses glycogen for itself and can make some precursors, but doesn't release glucose.
Right. Muscle is selfish with its glucose potential.
In a way, yes. Now, another absolutely crucial point, often a source of confusion. Fatty acid degradation yields mountains of acetyl-CoA. However, acetyl-CoA cannot be converted to glucose in animals. Let's hammer this home.
Cannot make net glucose.
Cannot make net glucose. Why? Because of the irreversible nature of the pyruvate dehydrogenase complex, PDHC, plus the 2 carbons that enter the TCA cycle as acetyl-CoA are subsequently lost as CO2 within that same cycle turn. There's no net gain of carbons from acetyl-CoA that can be withdrawn to make glucose. Instead, it's the carbon skeletons of most amino acids, the glucogenic ones, that can provide the building blocks and, of course, that glycerol backbone from triacylglycerols.
Okay, that distinction is fundamental. And linking back to enzymes, pyruvate carboxylase, PC, is the critical biotin-requiring enzyme, right? Carboxylating pyruvate to OAA.
Correct. That biotin cofactor is non-negotiable for that specific pivotal first bypass step. It enables the capture of CO2 to make OAA.
And when we talk about those unique bypasses, that conversion of oxaloacetate, OAA, to phosphenolpyruvate, PEP, using PPC, that really stands out as uniquely gluconeogenic, doesn't it, compared to the reversible steps?
Precisely. The conversion of OAA to PEP, involving PEPPK, is a reaction sequence truly unique to gluconeogenesis, clearly distinguishing it from the common reversible step shared with glycolysis. This two-step maneuver, PC then PPTK, is the clever bypass that gets around the irreversible pyruvate kinase step of glycolysis. And this brings us back beautifully to that concept of reciprocal regulation by AMP and fructose, 2-full-6-bisphosphate, which is just such a hallmark of elegant metabolic control. Both of these molecules inhibit fructose 1-full-6-bisphosphatase, slowing gluconeogenesis, while simultaneously activating phosphofructokinase 1, speeding up glycolysis. This elegant push-pull mechanism ensures that generally either glucose is being made or it's being broken down, but rarely are both pathways running full bore at the same time. It prevents those wasteful, futile cycles and optimizes the cell's energy use. It's fundamental metabolic efficiency.
Okay, so thinking about external factors, this raises an important practical question. What effect does something like drinking alcohol, ethanol metabolism have on this carefully balanced gluconeogenesis pathway?
That's a really important clinical connection. The metabolism of ethanol, primarily in the liver by the enzyme alcohol dehydrogenase, produces a large amount of reduced nicotinamide adenine dinucleotide, NADH, from its oxidized form NAD+ decision. This dramatically increases the NADH to NAD+ ratio within the liver cells. And this altered redox state has significant negative consequences for gluconeogenesis.
How so? How does the NADH mess things up?
Well, first, remember that OAA to malate conversion, high NADH drives that reversible reaction backwards, favoring malate formation and therefore decreasing the availability of mitochondrial OAA, which, as we know, is a key starting substrate after PC. Second, high NADH also drives the reversible reduction of pyruvate to lactate, catalyzed by lactate dehydrogenase. So it converts precious pyruvate, another key substrate, into lactate.
So it drains the pool of both OAA and pyruvate.
Essentially, yes. The net result of this flood of NADH from ethanol metabolism is a significant decrease in the availability of key gluconeogenic precursors, OAA and pyruvate. This strongly inhibits gluconeogenesis. This can actually lead to hypoglycemia, low blood sugar, especially in individuals who might already have depleted glycogen stores, perhaps because they haven't eaten or are malnourished. This is why drinking alcohol on an empty stomach can be particularly dangerous. It shuts down your backup glucose production system.
That's a very clear and important link. Okay, and finally, let's revisit that earlier, almost paradoxical question about acetyl-CoA one last time for clarity. Given that it absolutely cannot be a direct substrate for net glucose synthesis, why is its production from fatty acid oxidation still considered so essential for gluconeogenesis?
It's a critical nuance, absolutely, and mastering it really reveals the strategic brilliance of metabolism. While acetyl-CoA cannot donate its carbons for net glucose synthesis, its production from fatty acid oxidation is indeed essential because acetyl-CoA plays that critical regulatory role. It acts as a powerful signal. During fasting, when fatty acids are being broken down, the resulting high levels of acetyl-CoA inhibit the pyruvate dehydrogenase complex, PDHC. Remember, PDHC is the enzyme that would normally commit pyruvate to being burned for energy by converting it to acetyl-CoA. By inhibiting PDHC, acetyl-CoA effectively tells pyruvate, hold on, don't go down that path for immediate energy. We've got plenty of energy from fat. Instead, conserve yourself for glucose synthesis.
So it closes one door for pyruvate.
And simultaneously, this accumulating acetyl-CoA activates pyruvate carboxylase, PC, the first committed step of gluconeogenesis. It pushes pyruvate towards the synthesis of oxyl acetate and ultimately towards glucose.
While opening the door to glucose synthesis.
Exactly. So while acetyl-CoA doesn't directly provide the building blocks, carbons for glucose, it masterfully orchestrates the flow of other carbon atoms from amino acids, lactate, glycerol via pyruvate OAA to ensure gluconeogenesis proceeds efficiently when needed. It's truly a strategic metabolic traffic director, making sure precious resources are used appropriately for survival during fasting.
So wrapping this all up, what does this all mean for you, the listener? We've journeyed from understanding your body's dire, continuous need for glucose in certain tissues, to dissecting the specific molecules, those clever bypass enzymes, and the incredibly intricate regulatory mechanisms that make gluconeogenesis possible. We've seen how your body can remarkably turn components of fats, glycerol, and proteins, amino acids, into vital blood sugar, bypassing those irreversible steps in glycolysis with ingenious biochemical workarounds. And we've seen how it precisely controls this energy-guzzling process through hormones like glucagon and insulin, and through allosteric effectors acting as fuel gauges, like acetyl-CoA and AMP. It truly highlights your body's remarkable adaptive capacity and metabolic ingenuity. Really, a biological marvel designed to keep you functioning even when dietary carbohydrates are completely absent.
It really does. And this makes you pause and consider, doesn't it? Given the incredibly intricate, finely tuned, and frankly energy-intensive nature of gluconeogenesis, what does this tell us about the powerful evolutionary pressures that must have shaped our metabolic pathways? Especially thinking about surviving inevitable periods of food scarcity throughout human history. It truly encourages us to view the sophisticated balance between storing glucose as glycogen, utilizing it immediately, glycolysis, and synthesizing it de novo, gluconeogenesis, as nothing short of a masterclass in biological engineering. It's a testament to survival achieved through profound metabolic flexibility.
Absolutely, a profound testament to survival. But it also makes me wonder, maybe a provocative thought for you to consider. If our bodies are so brilliantly adapted to make glucose internally during times of scarcity, does our modern environment, often characterized by near constant carbohydrate availability, somehow short circuit or perhaps underutilize this ancient, finely tuned system? And if so, what might be the broader long-term implications for metabolic health? We certainly hope this deep dive has given you a profound appreciation for the elegant complexity behind how your body maintains its energy balance, even when you're not actively fueling it with carbohydrates. This understanding of core metabolic pathways like gluconeogenesis is truly fundamental to appreciating so many aspects of human physiology, health, and even disease. Thank you for diving deep with us today.